Everyone. How are you children? Doing well? Right. Am I audible? Yes. See, so I'm telling you the agenda of today first. After that, I will also say about what we are going to discuss from the next week. You can say from the Monday. So today, we will once again go for the detailed paper explanations of the NEET. After that, from the next week, means you can say day after tomorrow, we will start the track sheet and you will got the detail of the track sheets, right? So, but we will start in the very first week, what in the second week, so accordingly we will start the track sheet. So, patiently see the questions and what the purpose of the paper discussion, I already told you. The purpose of the paper discussion is you just manage to know where you did the mistake and why this mistake happened. You will find in many questions, you know the answer, but beside of that it become wrong. So you just try to identify that area, why it happened. Getting the things? So in the next NEET, we have to eradicate that mistake. We will not come, we are not going to do such kind of mistake again and again. Usually it's happened when you give the same paper after the examination to the students, their performance going to increase by 10% or 20% or sometimes 30%. Means what are the factors who stop you to perform great at the time of writing the real examination? And where you just find I have a lack of knowledge, I just got confused. So these are the reason we are once again discussing it. So listen it patiently. Discussing a paper what we given few week before is somehow boring, but beside of that, I will try to give you some knowledge along with the question also. So dear children, your 1.5 hours is not going to be a waste. Definitely, we will discuss some very important aspects along with the questions. Is it clear? So can you tell me up to which question we discussed in the last class? Anybody can tell me the question number? Right. Monday, what will be the first chapter? See, since Monday we will discuss some breach type of things where you struggle, like important terminology, important things which are going to help you to deal the biology in the different chapters. After that, we will just discuss about, we will start with the cell biology, right? So I think these are discussed. 129, 130, I hope it is discussed already, na? 131 probably also over. Anybody can recall up to which we have discussed? Less voice, is not audible very clearly. Hello, am I audible? Voice is coming good. One student is writing voice is somehow less. Okay, some are writing some cut coming. So just look on that. What problem is coming?
Their side internet problem, sir. Their side internet.
Okay, their side internet problem. No, all good. Okay, so check your internet connections, right? I think it may be an issue in the speed of the internet, right? So 133 we discussed. I hope not, na? So start from here. right? Okay, Ji. So look at these questions of this year NEET. Which of the following is not true regarding the release of energy during ATP synthesis through chemiosmosis? It involves, right? So have a look. Have a look here. The very first statement here is, the very first option here is breakdown of electron gradient. Is this option is correct or wrong? And why it is wrong? So you try to recall about the chemiosmotic hypothesis. I am giving you a brief synopsis of hypothesis, right? And you will understand as well. Look, so when you look the diagram of the NCERT, it is given like that, na? Here it is photosystem 2. Here B6F. Here who is there? PS1. And this one is what basically? CF node, CF1, and it is ATP synthetase. Okay, ji? These are the diagram given in NCERT in chemiosmotic hypothesis. Clear? Right. Now what happened? You just see, you will got a brief revision, fast revision of chemiosmotic hypothesis here. Whenever electron transport from PS2 to PS1, right, via B6F complex, right, what happened? Energy release. Okay, movement of electron occur downhill, and during that process what's happened? Energy release. And what these energy is doing? These energy is pumping the proton from where to where? Stroma to lumen. Okay, so the loss of the electron of PS2 getting compensated by what? Photolysis of water. Water going to split into hydrogen and oxygen, and from hydrogen electron is transported in PS2. So there is income up to proton here. Now when NADP plus converting into NADPH, proton requirement is fulfilled by which one? Stroma. Ultimately what happened? People unnecessarily confuse in chemiosmotic hypothesis. It is damn easy. See, this is the chemiosmotic hypothesis. What happened? After light reaction, proton concentration increase in the lumen. Earlier, a stroma and lumen have equal proton. Now, where is more proton? Lumen have more proton. How it become more in proton concentration? There are three methods for that. When electron move from PS2 to PS1, proton pump occur from stroma to lumen. Who lose? Stroma. Who gain? Lumen. When photolysis of water occur, income of proton occur. Where it occur? Lumen. And when production of NADPH occur, what happen? Proton loss occur in stroma. So overall, where is more proton? Lumen have more proton. Where is less proton? Stroma have less proton. What happened? Proton gradient will create, and proton return back to stroma. It's a line of NCERT. And when proton return back to stroma, what happened? ADP plus inorganic phosphate join together and form ATP. It performs conformational changes in the enzyme, and ADP and inorganic phosphate join and ATP produce. So this option is incorrect or not? Tell me, dear children. Incorrect or not? Hurry up. What happened? It is written breakdown of electron gradient. No, it is not the breakdown of electron gradient. It is breakdown of which one? Proton gradient. And that is the reason this option is a wrong one. Now, see the second option. Why you are just looking on the first option? See the second option. Movement of proton across the membrane to stroma. Yes, from lumen to stroma it is moving. It is correct. Reduction of NADP plus to NADPH2, it is also correct. After that, breakdown of proton gradient, of course, breakdown of the proton gradient, because one side proton is more, another side it is less. So all other options are correct other than first. So this is the detailed explanation of question number 133. Getting the things or not? Right? Okay, ji. Now, it may be next NEET exam, examiner will ask, which kind of proteins are present from where, through which proton is moving from a stroma to lumen? It is a pump protein because energy is requiring in that. Which type of protein are involving through which proton are moving from where to where? Lumen to stroma. So it is facilitated diffusion, carrier protein. Getting the idea? So there is scope of asking many questions. So if any student, why I am just giving the explanation, if any student have idea of this concept, they are not going to mistake in this question. Are you agree or not? If you know about this chemiosmotic hypothesis very comfortably, these are easy cake. These are the easy questions. Just these things I want to show you simply. Now come on the next. 134. I always say, in the very first class I told, examiner want to judge your patience. And how they check your patience? By asking the factual question. Information-based question. Questions which require mugging up activity. And that activity you don't like. And not one type of mugging up activity. Many type of repeated revision. A lot of patience required. That is the question to check your patience, dear children. What the question is there? No brain required to answer this question. It's a very factual question. If you revise the textbook 5, 6, 7 times, you will find, sir, what is the question? There is no standard in this question. If you not revise, there is a possibility you made it wrong. So what is the question? The flower are zygomorphic in. Answer it. Right? Zygomorphic flower. Actinomorphic and zygomorphic flower. Do you know na? Actinomorphic and zygomorphic. Actinomorphic is equal to which one? Radial symmetry. Zygomorphic is what? Bilateral symmetry. Where it is present? Cassia and Gulmohar. It is written in NCERT or not? Cassia, Gulmohar have zygomorphic flower. Direct line of the NCERT. And very easy question. Of course, member of family Fabaceae. Getting the things or not? I also have which kind of flower? If you go through the Fabaceae floral formula, it came like that, this symbol. So all the member of Fabaceae also have which one? Zygomorphic flower, but that option is not mentioned here. So pea, bean, cassia, gulmohar, such kind of example is given in NCERT or not? If you have a perfection in that, never you will do the mistake in such kind of questions. So simply checking of your patience. Now next. Just connect with me. Getting the idea. Otherwise, it is already paper done. You will find some boring work if you not got give a connectivity here. So now where the next question went? Just a minute. Give me a minute. It is done. 135. Hanji. Once again, checking of your patience. No IQ required in this question. Factual information. If any old student are listening me right now, I told many times, dear children, look after the, which one? Functions of micro and macronutrient. You will definitely got the question from here. I said or not said? Yes. And see, if you listen us very properly and follow that only, NEET, trust me, NEET become very easy for you. Because as a teacher, we know what are the important factual area from where question is going to ask. Simply you did it. What you do from the day one, you just take a register. That line I also said many times in the classes. You just take a register. Good quality of notebook. And accumulate all factual area together. And Example I am telling. Next year NEET also, there is also a definite question going to ask from this paragraph, from this particular paragraph of the NCERT. Getting the things? Problem is what? You people do simply crisis management. That is not required. Just manage yourself very properly. Getting the things or not? What are the area where revision require many time? That must be in your notebook altogether. And every day during study hour, 3 page, 3 page, 3 page, revise it every twice or 4 day or 5 day after. Getting the things or not? You got a great perfection. Never the mistake will happen. But all the things must be listed together with you. Are you getting the idea or not? Right? Next year also a question will come. And if you just listen from here, remove from here, okay, yaar, I will do it. Casual approach. Same mistake will occur next year also. So don't repeat it. Doing mistake is not a problem. Repeating is a blunder. Getting the idea, right? So, okay, let's come on the questions, right? Everyone know what to do, what to not do. Problem is what we are, if you are not doing, you will come in crisis. So do it. Well, collect the list. If you are finding difficult to collect, sir, what is important from here? Mugging up type of question going to ask to us. We will tell you, make a list of this, make a list of this, make a list of this, and mug up it. Definitely a question will come from every list. You got the idea? So now, how much easy question is it? Look here. How much easy question here is it? See the things. What is the question? Match the following. Manganese. What is the function of manganese? Manganese is associated with photolysis of water. Tell me other mineral along with manganese associated with photolysis of water. I am waiting for your answer. Write it. Very good. Chlorine. Manganese and chlorine both are associated with what? Photolysis of water. Very good. Very good. You did it correct. Second, come on, magnesium. Magnesium is act as an activator for enzyme of respiration. It is also very easy. Even you not mug up, you have idea about this step of the respiration. When pyruvic acid converting into acetyl coenzyme A, what happened? NAD plus converted into NADH, CO2 release, coenzyme require coenzyme A and magnesium ion. It is written in NCERT or not? These line is written in NCERT or not? Tell me, hurry up, hurry up. Even you forget from there, from here you can able to answer the question or not. So when you are thorough with the subject, usually mistake not happen. Got the idea? So honesty will pay you, commitment will pay you. It's not required to answer me or anybody else. Every day answer yourself. Today how much I did for my NEET. And this factual area, there is no scope of mistake. If you really very methodical, very honest, revising the things, getting the things very properly. Now, otherwise what happened? You will find your magnesium or manganese, who is doing photolysis of water, it will overlap in your brain. These things happen. And these small things is the reason behind we just struggled. Got the idea? Now, boron, pollen germination. Okay, ji, iron. Iron act as activator for enzyme catalase. Now match of the following. Answer came. Are you getting the idea? Is it all right? Now, and that is the reason I am just saying about every question. What the things required to handle this question. Either a concept required and it is a simply information, right? So you just realize what to do, what to not do, where what requirement is there. Now it is a conceptual question and it is an easy question. The very first line we taught, getting the things, to make the things simple. When I start to teach what? Plant-water relation. We say line na, if you talk about psi W, right, water potential, it decreases as you add the solute. As solute concentration increase, water potential decrease. How many times we said these lines in class, right? We say always, na? As solute concentration will increase, water potential will decrease. And I also told like that, solute concentration and solute potential are two different things. If solute concentration will increase, solute potential will also decrease. Water potential will also decrease. I think anyone who are listening me, our old student, check your notebook. That line you will find very directly written there or not. Many times we have given the questions, right? So how much easy question now? Addition of more solute in a given solution will lower the water potential. Right? It is a correct statement. Second statement, look, make water potential 0. No, make it correct. If you understand the concept, make it correct. Make its water potential 0. Is it correct or incorrect? It is incorrect, na? So come make it correct. What should you write so it become a correct statement? Make it water potential more negative. More negative. Got the idea? Not 0. 0 is the water. What happened? Pure water have maximum water potential, that is 0 at standard temperature and pressure. That's written in like when you add the solute. So the water potential of a solution is always negative. Are you getting the idea or not? Third option, not affect the water potential. No, it will decrease the water potential. It is written raise the water potential. No, lower the water potential. So who is the only correct option? You got the idea? So it is a easy question or not? Tell me. Unnecessarily we are increasing the blood pressure. Simply be methodical. Great will happen. Right? Next. (137). Haji. Oh, these are playing hide and seek. 137. Look. Read the following statement on lipid and find out the correct set of statements. See, first statement, tell me either it is wrong or right. Involve yourself. You already did the paper. Right, G? So, lecithin found in plasma membrane is a glycolipid. This is the wrong word written here. Lecithin is not a glycolipid. Then what is this? What is this? It is a phospholipid, not glycolipid, right? Second statement, saturated fatty acid possesses one or more CC bond. Correct or wrong? No, saturated fatty acid have single bonds, right? Saturated fatty acid have C, single bond C, not the double bond. Who have double bond? Unsaturated fatty acid. And anybody can tell me how many type of unsaturated fatty acid? I told when I teach, MUFA, PUFA, say or not say? MUFA and PUFA, mono unsaturated fatty acid, poly unsaturated fatty acid. PUFA, MUFA are unsaturated fatty acid. Now I am asking a small chemistry to you. Tell me, hurry up. Paper discussion doesn't means na ki question is there, answer is there. Okay, ji, work over. I am asking a small question. How can you convert a unsaturated fatty acid into saturated fatty acid? Who will answer it? Carry on. 9th class chemistry I asked. Unsaturated fatty acid go to saturated fatty acid. We have to perform hydrogenation. What we have to do? Hydrogenation. By hydrogenation, unsaturated fatty acid converted into saturated fatty acid. If you want to convert vegetable oil into vegetable ghee, you have to vegetable ghee. One of the popular brand name is what? Dalda. Getting the thing? Dalda is not a product, it's a brand name. It is a vegetable ghee. How to convert oil into ghee? Hydrogenation. Got the idea? Right? So, next statement, Ganglion has lower melting point, hence remain as oil in winter. Yes, oil have lower melting point than vegetable ghee. Getting the idea? Lipid are generally insoluble in water but soluble in some organic solvent. This is also correct. This is also correct. Getting the idea? Next, when fatty acid is esterified with glycerol, monoglyceride is forming. Yes, it is also a correct statement. But I am asking a question here. There is a scope of asking the question, right? So never left that scope when you are discussing the paper, right? This is the only knowledge gaining part we are discussing. In monoglyceride, tell me how many glycerol and how many fatty acid are there? Who will tell? Type your answer. How many glycerol and how many fatty acid are there in monoglyceride? 1 plus 1. Good one. How many ester bond there? How many ester bond? Priyanka and Taju have given the correct answer. 1 ester bond. In diglyceride, how many? In diglyceride, how many glycerol, how many fatty acid? Glycerol number in diglyceride? 2 plus 2. No. I am just writing. Just a minute. Give me a moment. Have a look here. It is damn easy. You will never got a confusion. Look here. Glyceride or glycerol and fatty acid is going to clear from right now. During paper discussion only, after that you will not got any confusion. That is a commitment. Have a look. What is this? See, I'm changing the color, it become more better. You tell me what is this. I hope many got in between before also. Just a minute, what I did. C, C, C. Here OH, 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 H, 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 H. What is this? It is the structure of what? Glycerol. Very good. What is the another name of glycerol? What is the another name of glycerol? Who will tell me? Trihydroxypropane. How many hydroxy group is there? 1, 2, 3. 3 carbon. Trihydroxypropane. Very good. So it is glycerol or trihydroxypropane. Now see what I am doing. I am writing here R1 COOH. It is a fatty acid. So when one fatty acid join with glycerol, what happened? Hydrolysis occur, a water will release. And glycerol and fatty acid join together by which one? Ester bond. Esterification occur. And then it is called monoglyceride. So one glycerol plus one fatty acid, right? Monoglyceride. One glycerol plus 2 fatty acid, what happened? Diglyceride will form 2 ester bond, and I am writing 3 fatty acid. It is forming a which one? Triglyceride. So in a triglyceride, in a diglyceride, how many glycerol are there? 1 glycerol, 2 fatty acid. In triglyceride, what is there? 1 glycerol, 3 fatty acid. So will you make a confusion? It is damn easy and it's an easy word. So glycerol, fatty acid, monoglyceride, diglyceride, triglyceride, number of ester bond, how they join together, esterification. Some basic idea came during discussion of question only or not? Tell me, hurry up. Clear. Right, Ji. Now we are moving ahead. Now we are moving ahead on the next one. Next question. What is the role of large bundle sheath cell found around the vascular bundle in C4 plant? Any student is here who did it wrong in the NEET? Tell me the honestly. Honesty is a great thing. Right now? It's somehow for a time being think like here what people will think about me. But you will feel very comfortable when you are saying very clearly. So everybody did it right. Okay, some students are writing. Honest one. Yeah, I did it wrong. Okay, no problem, no problem. So when you read the line of NCERT, NCERT wrote a line, कि the number of chloroplast in which one? Bundle sixth cell is more than the number of chloroplast in which one? Mesophyll cell. Right? That line, are you aware about, written in your textbook or not? Right? Number of chloroplast is in which one? Bundle sixth cell is more than the number of chloroplast in mesophyll cell. Right? So they are asking, what is the role of large bundle sixth cell? Okay, found around the vascular bundle in C4 plant. It accommodating a large number of chloroplast. And when large number of chloroplast is there, what happen? Who will operate better? Calvin cycle will operate better. So increase the number of chloroplast for operations of the Calvin cycle. Getting the idea? This is the correct answer. Okay, and in bundle sixth cell of the C4 plant, what happened? CO2 is going to pump with the help of enzyme. First oxaloacetic acid is forming, right? Then malic acid is forming. Malic acid enter in which cell? Bundle sixth cell, then converting into which one? Malic acid converting into pyruvic acid, decarboxylation occur, and during decarboxylation CO2 release. So effective pumping of carbon dioxide occur in which one? Bundle sixth cell. So the efficiency of a C4 plant to perform photosynthesis is better than which one? C3 plant. Now come on the next. Now come on the next question. These are playing hide and seek. Okay. Next question is, in your state, zoology teacher take care. It is in botany, but here in AP and Telangana, who take care of that? Zoology, sir, because ecology in your state in zoology. Okay, which question is next? 140 question. It is a damn easy question, right? If any old student are listening, I say only thing, ki when you ask the student, tell me what the problem in application of biology. Students say, sir, RNAi. Right? You say, na, RNAi is looking difficult, sir. Always I say, 4 line, 5 line of RNAi, if you keep in your mind, never you make a mistake from RNAi. Once again, I am repeating this line here. And it will also applicable for the next NEET. Got the idea? I am saying 5 line about RNAi. In NEET, no one asks more than that. That one have to keep in mind. Unnecessarily, there is no need to complicate the thing and make it difficult. Right? Listen it carefully. I am saying about RNAi. What you keep in the mind, note it in your mind. So look, RNAi. What is RNAi? It is a method of cellular defense. Keep in your mind. What is RNAi? It is a method of cellular defense. In which type of cell? In eukaryotic cell. Second point. In eukaryotic cell. How? By post-transcriptional gene silencing. So during RNAi, who is not going to stop? Transcription is not going to stop. Who is going to stop? Translation is going to stop. Till now, it is okay or not? So RNAi is a method of cellular defense, right? In which cell? Eukaryotic cell, not in a prokaryotic cell. Why? Because in prokaryotic cell, regulation of gene occur only at transcription level. But in eukaryotic cell, you studied the 4 level of regulation is there. So RNAi, we are stopping the translation. So it is a method of cellular defense in eukaryotic cell. Now another question examiner asked from RNAi is what? I am giving you a brief idea of RNAi. So next NEET, no one even ask more than this. If they are asking question from the RNAi, is it clear? So next slide. During RNAi, what is the source of nematode specific gene? Who will answer? This is also a very important line from RNAi, from where question is going to ask. RNA virus, first, second, transposome. It is written, and this is the question. कि transposomes can be used during which of the following? Gene silencing. RNAi is a method of which one? Gene silencing. How? To stopping the translation, to not allow the gene to express a protein. Able to understand? And another thing examiner may ask, what are the vector used during RNAi? Agrobacterium. And what another important line you have to keep? We transfer the nematode specific gene in such a way it produce a double stranded mRNA. You got the idea? Is it done? So keep that much information about RNAi. You will never struggle from here in the NEET exam. Now next. Match the following. How much easy question? How much easy question? Right, G? 141 is very easy, na? Where it went? 141. They are asking about what? Chromosome and related terms. Metacentric chromosome. This classification of chromosome are based on position of centromere. So when it is how much easy? You better know. There is no scope to do wrong here. If who is in center? Centromere is in center. It is metacentric. Somehow away from the center. One arm is long, one is somehow short. Submetacentric. One very short and another very long. Acrocentric. Only single arm. Terminal centromere. It is which one? Telocentric. Are you getting the idea? So these are the 4 type of chromosome. Here, when you look the shape, it will give V shape, it will give L shape, it will give J shape, it will give I shape. Now I am asking a question. In which stage you study the shape of the chromosome? Suppose that next NEET a question came. We study the shape of the chromosome in which stage? Just a minute. Prasad ji. In class, all done. Carry on, Ji. Hurry up. Anaphase, good one. We study the shape in which one? Anaphase stage of the cell division. It is also a very potential line for examination. Right? So it is also over. Now we are going to move on the next. And what is the next? Next. It is also an easy question. But if you talk to student, they somehow struggle. Why I can't say? Wait a minute. Hanji, this question came now. Match the plant with the kind of life cycle. See, if you look the trend of this year NEET, they really frame the question in a proper manner. If you have a thorough knowledge, then you can answer the question only. This is the beauty of a statement-based question. So this time, paper setter taught you something. Ki don't take any subject as granted. Don't always think like I will save the question of biology and I will use in physics. I hope for many students, biology have taken some time of physics also. It's happened or not? That much long statement. Right? So how they just kill your strategy, you got the idea? So never take any subject as a granted. And see, trust me, it is not a difficult paper, it is a lengthy paper. Right? So any student who find like, okay, I will do biology in that much minute, I will use that time in physics, their entire strategy gets spoiled. Any can do this question in due time, but one condition is there. Your reflexes must be great. Reflexes, how it become great? Always my favorite question. 2 2 ja 4, 13 8 ja 104. I have taken a pause when I was thinking 13 8 is 104. 2 2 reflex 4. So if things at that level of perfection, even you can do such things in very faster way. Now come on the life cycle. These, if any student have idea about all the life cycle given in NCERT entire paragraph, they did it very correctly. If you struggle, you just find difficult to do it. So once again, I got an opportunity to teach the life cycle in 1 minute. Teacher got the opportunity to say something during the paper discussion. So I got the opportunity to explain the life cycle in 1 minute or 2 minutes. Listen me. I am telling about all life cycle. In NCERT, how many type of life cycle is given? Heplantic, Haplodyplantic, and diplantic. 3 type of life cycle is given. I am not looking at the question. I am not giving the answer. You will do it now. I am just telling you the life cycle. How much information required to answer this question? I am teaching you that. And after that, you do the question yourself. So how much information requiring? Listen, Heplantic life cycle. Gametophyte is dominant and independent. Agree? A sporophyte is parasite on gametophyte. A sporophyte don't have an independent existence. Such kind of life cycle is Heplantic life cycle. Where it is present? Heplantic life cycle is present in most member of Thallophyta. Thallophyta means what? Algae. Heplantic life cycle. But some algae are in the exceptional condition who have a diplantic life cycle also, haplodiplantic life cycle also. Fucus have what? Diplantic life cycle. Polysiphonia have what? Haplodiplantic life cycle. Beside of that, they are algae, but most algae have what? Heplantic life cycle. Right? Done? Over. So you got the idea about the heplantic life cycle? Now, haplodiplantic life cycle. These are present in 2 groups, Bryophyta and Pteridophyta, according to NCERT. So haplodiplantic life cycle in Bryophyta, what happened? Listen it carefully. You got a brief synopsis of the entire life cycle in 2 minutes, right? So in haplodiplantic life cycle, gametophyte is dominant and free living and independent as well. A sporophyte is either parasite or semi-parasite on gametophyte. Sporophyte don't have independent existence. They are dependent on gametophyte for their existence. Such kind of life cycle present in members of Bryophyta. Clear or not clear? Done or not done? Right? If you have studied this, they came in a flashback in your mind. Yes, this paragraph over. Now third, haplodiplantic life cycle also present in Pteridophyta, according to NCERT, right? So in Pteridophyta, what is the story? In Pteridophyta, sporophyte is dominant. But both sporophyte and gametophyte have independent existence. And gametophyte is what? Prothallus. That is in the case of which one? That is in the case of which one? Pteridophyta. Pteridophyta is the only peculiar group where both the stages N and 2N stage have independent existence. They not depend on each other. Clear? Now come on gymnosperm and angiosperm. One type of life cycle is there. What the name? Diplantic life cycle. Now what happened in the case of diplantic life cycle? A sporophyte is dominant and gametophyte is reduced and parasite on which one? A sporophyte. Over. That much information only required to answer this question. Means one page, entire page of the NCERT. Now you better know, a Spirogyra. It is an algae. Which kind of algae is it? Tell me, hurry up. Answer it. A Spirogyra is green, brown and blue. Sorry, red algae. Green algae. Fern. It is a pteridophyte. Cycas, gymnosperm. Funaria, bryophyte. So dear children, today I am giving you a small task. You do it by today. Should I? Can I say something to do? Are you ready? Yes. So you take a notebook today, and in your notebook, in plant kingdom, whatever the example is there, you collect at one place. How? You just give a heading, examples of plant kingdom. Green algae. What the name of green algae given in NCERT? Write all together. Brown algae, red algae, bryophyta. Who is liverwort? Who is hornwort? Who is mossage? Pteridophyta, gymnosperm, angiosperm. You just accumulate all example together. It will help you to make the confidence in this area up to many level. Getting the things or not? Your confidence is going to increase. Otherwise, student struggle, it is brown algae or red algae. Tell me, polysiphonia is a green algae, brown algae or red algae? Polysiphonia. Porphyra. Hurry up, rapid fire round. Porphyra red. Very good, Janvi, very good, Mohit. Right? Right. Ictocarpus. Carry on. Brown algae. Volvox. Brown algae. Kelp. Volvox, green algae. Right, very good. Right, very good. So maintain these perfection. Got the idea? Volvox is green algae, kelp is brown algae. Right? Very good. Keep it up. Next. Next question is, which of the following fruit labeled in the given figure makes a false fruit? Thalamus, when it is edible, it is a false fruit. In which fruit you eat the thalamus? Apple, pear. Getting the things or not? We eat which one? Thalamus. So it is a very easy question. C part. Next. 146. How much easy question is this? You find like any question more easy than this question? Anybody will give you more easy question, right? Na? So it happening na, when you did the paper, it is looking like here it is too tough. When you are discussing it, you are finding I know everything. What but big there? Able to understand it is damn easy. Okay, ji. In the following palindromic base sequence of DNA, which one can be cut easily by particular restriction enzyme? Getting the things. Who is palindromic sequence here? Who is palindromic sequence here? Palindromic sequence is what? Forward and backward directions, reading frames are same. I am writing GAATTC. I am writing here 5 prime to 3 prime. Here also anti-parallel 5 to 3 prime. I am writing GAATTC. It is a palindromic sequence. This is the example, only example given in NCERT, right? So this palindromic sequence going to cut by which enzyme? Who is going to cut it? EcoR1 is going to cut it. Which enzyme is cutting? EcoR1 is cutting. Getting the things or not? Is it clear? Right? Other than this, no one is a palindromes. Right? Other than this, no one is a which one? Palindrome. Now I am asking another question. Okay? GGATCC. GGATCC. Is it a palindromic sequence or not? Is it a palindromic sequence? Yes. Now I am asking another question. Which enzyme will cut it? This sequence getting cut by EcoR1. Who will cut this sequence? Anybody can answer? Answer it. Who will cut it? It get cut by very popular enzyme. You are studying in your NCERT, but it is not written. Keep in mind. Discussion means we also give you some information, na? Then it is birthful, na? Otherwise it look boring, na? All the class. When you are discussing even the old paper, you got some relevant related knowledge. This is the purpose, right? So when you are listening someone, you must got something, output must came. So BAM H1, will you keep in mind, will you forget? BAM H1. Clear G? Can we move ahead? This is why eco R1. BAM H1 you use na? In NCERT, which enzyme they are using in PBR 322? This one na? And this is going to cut this particular palindromic sequence. It's over. That's an easy question. Now next. It is how much easy question. Which of the following will accelerate phosphorus cycle? What is the reservoir of phosphorus? Rocks. When withering of rock will occur, what happen? Phosphorus will release in soil na? And cycle get accelerated. Getting the idea? Is it clear? Now next. 149. Simply they check your knowledge of linkage and independent assortment. So I am telling you about linkage and independent assortment. Question is going to solve automatically. Listen me. Listen my few word about linkage, my few word about independent assortment. Then your problem will sort out. This question become an easy cake. 3-4 line listen. Keep one thing in mind. Linkage is only shown by two genes present on same chromosome and close to each other. Linkage is never shown by two genes present on different chromosome. So what the first condition for linkage? Two genes must present on same chromosome. Okay, Ji? Two genes must present on same chromosome, A gene, B gene. They are present on same chromosome and close to each other, nearby of each other. They will show what? Linkage. Clear? Done. Now, linkage means what? When two genes inherit together in next generation. When two genes inherit together in next generation, it is called linkage. And for that, what is the condition? Two genes must present on same chromosome. And only presence on same chromosome is not sufficient. What are the other things required? They are close to each other. Now, when two genes present on different chromosome, they always show independent assortment. They never show linkage. And even when two genes present on same chromosome, but far from each other, they may show independent assortment. How? How? By crossing over. Wait. Here it is not. Here it is right. By crossing over. So, in independent assortment always shown by two genes present on different chromosome. Right? If two genes present on same chromosome, they may show linkage, they may show independent assortment. When show linkage? When they are close together. When they show independent assortment? When they are far from each other. And crossing over possibility is there. That much information, dear children, required to answer this question only. And every teacher deal it, I hope. If any old student are listening, I said such kind of words in class or not? Tell me. You can check your notebook as well. Right? Okay, ji, have a look. Now, answer came or not came? Now answer. Assertion. Mendel law of independent assortment does not hold good for the gene that are located closely on the same chromosome. True or false? Law of independent assortment not occur when two genes are on same chromosome and close together, they show linkage. So they are telling does not hold good. So the statement is a correct statement or incorrect statement? Tell me, hurry up. True statement. Very good. Next, closely located gene assort independently. True or false? Closely associated gene not assort independently. It is, they show linkage. So second statement is a false statement. So if you have that much information, you find like very comfortable to answer that question, ki sir, it is easy one, I can handle it. So dear children, confidence is the most important things in exam. You know the subject, I also know it. You studied well also, I also know it. Only what the things happen due to that reason you are here, we also note it. And I am telling you also why you are here. Reason being, what you know, that not with a great perfection level. Reflexage was slow. You not revise, optimize number of time what required to develop a great reflexage. You just taken more time in some of the question because that things was not very properly revised to you. And as you are taking more time in one question, your psychology, your pressure increase, your anxiety increase, how to handle the question. Right? You came in hurry, start doing the things wrong. One of the reason. Another reason, with lack of revision, not come with a perfection. With lack of revision was there, so you just struggle. How you struggle? Your manganese or magnesium in photolysis of water. Such type of things overlap only. You have to make it very clear. And where a conceptual mistakes are there, we are there for you. You ask as many times, sir, what you told, I don't got it. Tell me once, twice, thrice, as many times. Getting the things, as long as confidence not generated. Right? Okay, ji, next one. Next one. 150. And 150 is not the last. Then I will go in zoology also. 150 in which one? 150 is associated with zoology, sir. Actually, what's happened? In your state, what we are teaching, zoology, sir, taking care of some part of botany. Botany, sir, or ma'am, taking care of some part of what? Zoology. So I am coming on second part of the zoology. Some question of botany is also in zoology. So I am going in that area. Okay. So have a look. It will require time to search it, but don't worry. We will search and we will do all the question. If you are doing anything, do it very amicably and honestly. Patience required. I told na everywhere patience required, and it is your first testing of the patience. We are discussing a paper. You wrote a paper already. I know it looked like somehow boring. To revise the same paper once again what I did few days back. Haji. So you will help me? Okay, very good. So 153 question number also we told. 153 question number from which chapter? Reproduction in organism. Who taught you? Botany, sir. Right? Okay. Reproduction in organism. So they are asking, identify the asexual reproductive structure associated with penicillium. Penicillium belongs to which class? Hurry up. Ascomycetes. In ascomycetes, asexual reproduction occurs by which method? Conidia formation. We told or not? I even told the life cycle of conidia formation. Asexual life cycle. Told or not? You see, it is conidiophore. This is conidia. Conidiophore. It is what? Exosporic germination occur, not a, here who will form? Conidia, it will germinate and after germination form new mycelium. Told like this or not? Told like this or not? Told na? Right? So easy question, 153 done. Now we are moving on. You tell me the question number if question bank is with, paper is with you. Otherwise it required to search. One student are saying 158, 161, 167. So come on 158 first. 155, 55, 55, yeah, it is problem creating and dragging. Oh, 61 came. 58 you told, na? Ha, 58, good one. How much easy question is it? It is easy or not easy? Tell me, hurry up. Easy, na? Breeding crop with higher level of vitamins and minerals or high protein and healthier fat is called what? Biofortification. Plant breeding to enhance the nutritional quality of the food is called what? Biofortification. Right? Okay, ji? Very good. So this is the answer. Next question, what you wrote? 161. 161. Which of the following statement with respect to endoplasmic reticulum is incorrect? SER is devoid of ribosome. It is a correct statement. You know, na? Endoplasmic reticulum is of two types, RER or SER. RER, rough endoplasmic reticulum, that have ribosome. ACR don't have ribosome over it. In prokaryote, only RER are present. Wrong statement. In prokaryotic organism, no any membrane bounded cell organelle are present. Clear? Endoplasmic reticulum is a single membrane bounded structure. No any membrane bounded structure present in prokaryote. So who become wrong? Second become wrong. Answer is available. ACR are site of lipid synthesis. Yes. Lipid and steroidal hormone, two line written in NCERT or not. And then RER has ribosome attached to ER. It is also correct. So all are correct except 7. So 161 is done, dear children. Now it is 167 you are saying. Okay. '62, '63. It will go up by hand. Yeah, now it okay. 65, 66, 67. Yeah. 167. Look. Select the incorrect statement with reference to mitosis. A spindle fiber attached to centromere of the chromosome. This is a statement which is incorrect. How? How it is incorrect? What is the correct one? Kinetochore. Don't confuse. Anyone have confusion between centromere and kinetochore? If you will say yes, I will make it clarify right now. No one have confusion, na? Kinetochore and centromere, then good. I am not discussing it. Okay, ji? Right. Done? Next question. This is a blunder done by NTA. Right? It is a wrong question. Do you know how it is a wrong question? They have asked the question ascending order. And every option is given in descending order. No any option is given in which order? Ascending order. Are you getting the idea or not? They have asked ascending and they have given all option in descending. So now you do it. It is a wrong question framed by the NTA. Got the idea? So this is the unfortunate of the country. In a single year, people are not able to frame a correct question paper. Agencies dedicatedly for that work, and just even mistakes are there. If people are making 20, 30, 50 paper, we can understand human error are possible. But a national level exam, it looks somehow disappointing. Okay, anyhow. So what is the sequence? Kingdom, phylum, class, order, family, genus, species, everybody know it. But they just tortured you by asking the wrong framing, getting the things. Name, next question, 169. Why I am doing from here? 169. Given below are two statements. Okay, let me shrink somehow. Given below are two statements. Statement 1, restriction endonuclease. Here spelling errors are there. Restriction endonuclease recognize a specific sequence to cut DNA known as palindromic nucleotide sequence. Tell me, correct or incorrect? Correct. Restriction endonuclease recognize palindromic sequence. What is the second statement? Second statement is restriction endonuclease cut the DNA strand a little away from the center of the palindromic site. It is also correct because NCERT only wrote about staggered cut. In NCERT not wrote about which cut? Blunt cut. But they may cut at center. But they may cut from away from center. NCERT author wrote which cut only? Staggered cut. They shown you as an example eco R1. And in eco R1, which kind of cut is going to occur? Staggered cut. So it is also looking correct. Able to understand? Okay, Ji. So both the statements are correct. Now come on the next question. Next question. Given below are two statements. First statement, mycoplasma can pass through less than 1 micron filter size. Correct or incorrect? It can pass through the bacterial filter. Okay, mycoplasma are bacteria with cell wall. No. Second statement is wrong. Mycoplasma don't have cell wall. These are bacteria without cell wall. Got the idea? They don't have cell wall. And that is the reason mycoplasma is called what? Joker of plant kingdom. Some people also say what? Joker of microbiology. They are pleomorphic. Change their appearance. Right? Next question. Next question. Which one is there? Okay, let me search. 174 is there, which is from the botany side. Identify the microorganism which is responsible for production of which one? Immunosuppressive agent. How many times I told in class? Revise it also. Ask you to repeat it also. Told these three are very important name. Said or not said? Yes, na? Immunosuppressive agent, blood cholesterol lowering agent, and clot buster. Told or not told? Told na? Many times. Question reflected or not reflected? Reflected. Right? Okay, ji. So, identify the microorganism. Tell me the answer. Immunosuppressive agent. Who is there? Immunosuppressive agent. Trichoderma polysporum. Getting the things. So it is easy one. All options are there. Next one, next one, next one. It is zoology. It is botany. And it is a very good question. Very good question. Impressive question. Right? You did it correct? 176 or not? 176, you did it correct or wrong? Correct. Good, good, Shreya. Good one, good one, good one. Did it. Very good. It's a very impressive question. What is the question? Let me tell you. In an E. coli strains, I gene get mutated. Mutation occur in which gene? I gene. So look here, I am drawing a diagram to make you understand the things in a better way. It is promoter for repressor. Here it is repressor, regulator gene, promoter for regulator gene, I gene. Here it is which one? Here it is promoter, operator, a structural gene, ZYA. Now they are telling I undergoes in mutation. I gene undergoes in mutation. So when it undergoes in mutation, what happens? A different type of protein will produce, na, or not produce. So here it is repressor protein. A repressor protein who is there is not forming. Same protein is not forming. Either no protein is forming due to mutation, or what happened? Different protein is forming. So, but these are not the repressor protein who bind with which site? Operator site. Got the idea? And its product cannot bind to the inducer molecule. Who is the inducer? If growth medium is provided by lactose, what will be the outcome? What will be the outcome? Tell me. What will be the outcome? Tell me, hurry up. Answer it. What is there? Regulator gene undergoes in mutation and not binding with which one? Inducer. Repressor protein and inducer not reacting together. Then what happened? Usually what happened? Repressor protein, getting the things or not, is going to bind with the inducer, which is the lactose. And when it is going to bind, what happened? Carry on. What happened when it is going to bind? They get inactive. They get inactive. Now they are not getting inactive because they are not able to bind with which one? Lactose. Now they are not going to inactive. And when they are not going to inactive, what happened? It remain active. And when it remain active, what happened? Bind with whom? Operator. Promoter for regulator gene will not bind. Process get off. Got the idea? Right? Next. Next, which one is? Okay, hand one is working now. Huh. Which of the following is a correct statement? Bacteria are exclusively heterotrophic organism? No. Some bacteria are also autotrophic. Next, slime mold are saprophytic organism classified under kingdom Monera? No. It is in Protista. Mycoplasma have a DNA, ribosome and cell wall. DNA is okay. Ribosome is okay, but don't have cell wall. See, 2 questions came from mycoplasma, cell wall based. Cyanobacteria are group of autotrophic organism classified under kingdom Monera. It is correct. Now, come on next question. You did it correct? 188 question. 188 question. How many of you did it correct? Easy question from N15 and N14. It is easy question, na? 10 E. coli with N15 double stranded are incubated in medium containing N14 nucleotide after 60 minutes. So see, 1 E. coli you can see. 1 E. coli you can see. N15 medium. 60 minutes means how many cycle? 3 cycle. 20 minutes, 40 minutes, and 60 minutes. After 60 minutes, how many bacteria? 1 to 2, 2 to 4, 4 to 8 bacteria. 1, 2, 3, 4, 5, 6, 7, 8. Out of 8 bacteria, in 2 bacteria, 1 strand are N15. Rest all the strand are which one? N14. So these are the story after which one? 60 minutes. After 60 minutes, out of 2 bacteria, out of 8 bacteria, 1 to 2, 2 to 4, 4 to 8. Out of 8 bacteria, 2 bacteria have N15 strand. Now what they are asking the question? 10 E. coli level with N15 double strand are incubated in a medium containing N40 nucleotide. Means growing in normal medium. After 60 minutes, how many E. coli cell have DNA totally free from N15? How much percent is this? How much percent of this? 6 upon 8. 6 upon 8 are free. 6 upon 8 are free. 6 upon 8 are free. Getting the things or not? Out of 8, N15 is not present in how many bacteria? 6 bacteria. Now, when it is 60, how many total bacteria we started? We started total how many bacteria? 10 bacteria. In place of 1 bacteria, here you have to 10 bacteria. Out of how much bacteria? 10 bacteria, 1 bacteria convert into 8 bacteria. 10 bacteria convert into how many bacteria? 80 bacteria. So 6 upon 8 of 80 bacteria, how much is it? 67. This is the question. Out of 80, how many? 60 bacteria don't have what? N15. Right? 6 upon 8 are normal. Able to understand or not? Next one. Next one. You did it correct, 194. It is easy question from the gene mapping. First, what the tricks you have to apply? See who is the longest one. So they are telling A and D is how much percent? I am writing here A, I am writing here D. They are asking 29%, 29 map unit. Right? It is 29 map unit. Now they are asking A and C is 5% and B and C is 15%. Right? B and D is 9%. Okay, Ji? A and B is 20% and C and D is 24%. Right? A and D is 29%. So whatever is the percentage, you just write like simply and go for a gene order. A, C 5 map unit. B, C 15 map unit. B, D 9 map unit. A, B 20 map unit. 20 map unit. C, D 24 map unit. And A and D how much? 29 map unit. Now you have to go for the arrangement of the gene. Got the idea? Time up. Okay. So are you in position to handle it? How many of you make it correct? How many of you make it correct? Simply you have to go for a small way out. How to handle this question? I am giving you an idea. You can handle any question. You have to go for different permutation combination. I am giving you an example through which you can handle such kind of question. Suppose that AB given 15 map unit, BC given 10 map unit, AC given 5 map unit. What is the sequence? So always keep a middle word. Suppose that I am keeping A, B, C as a sequence. Let me check either it is correct sequence or wrong sequence. AB given 15, BC given 10. So what is the AC? AC is 25. In AC here it is given 5. So this arrangement is a wrong arrangement. You have to check like this. Now I am telling you the process. That is the reason I am telling the longer way. Now here I am writing middle one A. I am writing BAC. Right, check either correct or BA 10, AC 5, BC 15. Getting the things or not? BA 10, sorry, BA how much is given? BA given 15. BA given 15. AC given 5. So what happened? BA 15, AC 5. So BC 20. It is also wrong. Here BC coming 10. Now what happened? I am putting C in the middle. So B, C, A I am trying. So BC given 10. AC given how much? 5. So AB given how much? 15. And AB also coming how much? 15. So this distance and this distance is matching. So you have to go for this permutation combination, the answer will come. It is easy or not easy? I did it all wrong method first. First I did this wrong method. Then I told wrong one. Then lastly I come on right one. So you can got the idea how to come on the right answer. So in such a way, you can just go like, but what the tricks you have to follow? First go for the longest distance. It will help you to find out the another one and make a relation of one with the another. Answer will come. So is easy question or not easy question? You do it by yourself. Okay, done, over. Any more is there? I think it all over. Thank you very much. It is zoology, 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 zoology. It is over. Thank you very much. So from next class, we will come on the topic and we will send you the track sheet also, what we are going to deal. Okay, ji? Thank you very much. Bye-bye. Have a good day to all of you.